Hello everybody, we are going to get started on chapter 10, muscle tissue. So we figured you're probably pretty sick of bones by now. Let's switch to muscles. I want you to make sure that you understand that we are not requiring you to know the hundreds and hundreds of muscles that are in the body. What we require you to know is listed in your lab. And so when you start looking at muscles and you think, oh my gosh, I can't learn 640 plus muscles, you don't have to learn that many. So there is a specific list, and in that list you need to know the muscle, how to spell it, the origin, the insertion, and the action of the muscle. So please refer to your lab guide to know which muscles you have to learn. Now, there may be some questions. The lab guide is actually on lab in the lab master. It's not a printed copy, but you can feel free to print any part of it that you would like. Okay, so just trying to do some housekeeping there in the front of this lecture. So when we start talking about muscle tissue, you should remember that this is one of the four major muscle types. You got to view this in lab, and of the three types, of course, you have cardiac, skeletal, and smooth muscle. The one thing that's important about when we start studying muscles, the only muscles you're going to identify, first of all, are all skeletal muscles. So that's important to understand. There is only one cardiac muscle, but of course there are millions of smooth muscle if you look at the cellular level. As we start into the subject of muscle tissue, you have to understand that muscle is very specialized because it has the ability to actually contract, and that is unique to the muscle tissue. You do get into some transitional epithelial tissue that can expand, and but that's not quite the same as contracting. And the reason that contraction is important is because it enables the muscles to contract to move the bones or the skeletal system in order to provide movement. So this is extremely important. Common properties, I have those listed in your lecture notes, but the muscles can respond. This is called excitability. They can shorten or contract, so they have contractibility. They're able to stretch, which is called extensibility. And finally, they're able to recoil after they've been stretched out, and this is known as elasticity. These muscles should look very familiar from your chapter on the study of tissues and from your lab exercise on studying tissues. One thing you should remember about when you start looking at types of muscle is whether or not it's striated. Of course, the only one not striated is smooth. Whether or not it's voluntary or involuntary. The only voluntary is skeletal muscle. And you should also know where the types of muscle are found in the body, because that's going to relate to function. So what are the functions of your muscles? Well, first of all, movement, okay? Without muscles, those bones are just a bag of bones, so to speak. You're not going to be able to move. Muscles are important for maintaining an upright posture. Without muscles, you would not be able to actually walk in an upright fashion. So those muscles in the rector spinae muscles keep your posture in alignment. Muscles also provide support to the entire body. They also support and help protect your internal organs. Muscles guard entrances to and from your body. Think of your sphincter muscles. And then muscles are really, really crucial for maintaining body temperature. Lots and lots of the energy generated by muscles actually is expended to keep the body at a specific temperature, and that would be 37 degrees centigrade. And finally, muscles help store nutrients. And we don't usually think about that, but muscles can actually store glycogen, which is the stored form of glucose, as well as some other nutrients. So these are important to understand. As you're looking at muscles, you should know types of muscles. You should be able to compare and differentiate these muscles. You should know the functions of muscles. You should also note the fact that muscles are not just made of muscle. No pun intended there. So when you look at muscle, specifically we'll start with skeletal muscle. If you were to take and dissect out that muscle, you would find connective tissue, you would find blood vessels because muscles have to be nourished, and you would find nerves. And nerves are very important, and we'll talk about that later. So here we can see the 3 layers of connective tissue that enclose the separate parts of a muscle. Now, this looks very similar to the nerve. If you remember looking at nervous tissue, instead of epimysium like we see in the muscle, you would have seen the epineurium. The reason I point this out is because if you already know epi, peri, and endo, you can simply change the root word from neurium to mysium, and now you've got the layers of the muscle. So when we look at the epimysium, this is going to enclose the entire muscle, and it's going to separate or insulate the muscle from the surrounding tissue. The next layer that we see is the perimysium, and this is going to surround the fascicles, and the fascicles are actually bundles of muscle fibers. Now, it's important to note that a muscle fiber is the same exact thing as a muscle cell. Those words are used interchangeably. The last layer that you see is going to surround the individual muscle fibers or muscle cells, and this is the endomysium, so the endomysium. The other thing that you can see here is the tendon. So this white connective tissue that you see running into the epimysium is the tendon. Now, it's important to understand that the 3 layers, so the epi, the endo, and the perimysium, all come together to form the tendon, okay? And so that tendon is what is going to attach the muscle to the bone. Okay, so tendons attach skeletal muscles to bones. It is not a ligament. Ligaments attach bones to bones. Tendons attach muscles to bones. A lot of people get that confused. Let's take a look at the muscle fiber or muscle cell individually. If we look at this, we can see that it's striated. It has many nuclei in the mature muscle fiber. But before we get to the mature muscle fiber, how do these start out? So we start out with what are called myoblasts. And myoblasts are simply the fusion of your embryonic cells. So those embryonic cells turn into myoblasts. The myoblasts are going to develop into the immature muscle fiber, and you can see that you've got the multiple nuclei and you're starting to see some of the striations. The other really special cell that you encounter associated with the mature muscle fiber and even the immature muscle fiber is the myosatellite cells. And these cells are actually a special stem cell that helps repair the muscle. This is a slide you probably are somewhat familiar with if you remember from Bio 205 and looking at the tissues in lab. Notice the striated appearance of the skeletal muscle. Also notice the black nuclei. You have hundreds and hundreds of nuclei within the skeletal muscle, so they are multinucleate. The other thing that you have to understand about skeletal muscle is it has a large vascular network, and so you can get a lot of blood and a lot of nutrients and oxygen going to the muscles, and this is important for your sympathetic nervous system, fight or flight. The other thing that's important to remember in the skeletal muscle is it only contracts when it is told to do so by the central nervous system. So you probably remember that from studying the nervous system. And finally, the skeletal muscle is the only muscle that is voluntary. And there's one caveat to this. It's still voluntary, but we don't think about it so much, and that is the diaphragm. So even though the diaphragm is is skeletal muscle, we don't always think about the fact that it's contracting. So it's still considered voluntary, but it's kind of a gray area there. When we looked at the layers of the muscle, we looked at a tendon. So all the layers came together to form the tendon, which attached the muscle to the bone. Now I want to look at what is called an aponeurosis. So as you look at the external oblique, you see this sheet, and this is what is called a tendinous sheet. And what it does is it serves as the insertion or origin for a skeletal muscle. So instead of a tendon, you have a sheet, which is called an aponeurosis, and you can see that on the bottom left. You can also see on the bottom right that you have the inguinal ligament, and this is at the border of the aponeurosis. So the ligament is completely different. What you're looking at is that sheet of connective tissue. So this is an important definition, and you need to know the difference between the tendon, which is a bundle, versus a aponeurosis, which is a sheet of tissue. So let's look at the parts of a muscle cell. They are given special names unique to muscle cells, but they correspond to the components we see in all cells within the body. And we're going to start with the outer layer, the plasma membrane. It is called the sarcolemma. Interior to the plasma membrane, so on the inside of that plasma membrane, it is going to surround the cytoplasm of the muscle cell, which is known as the sarcoplasm. And this is the cytoplasm of the muscle cell. When we get into the further into the cell around each individual muscle fiber, so on this slide we're looking at a fascicle, and then if we look at the individual muscle fibers, we can see that they are surrounded by these blue, what are called sarcoplasmic reticulums. We shorten that to SR because these are very similar to the ER, endoplasmic reticulum, that we would find in a regular cell. So the sarcoplasmic reticulum is delineated, so on each end it has what are called T-tubules. And these T-tubules are transverse tubules. And these transverse tubules are really important because they are going to take the action potential and carry it deep within to the muscle layer itself. At the end of each of the sarcoplasmic reticulums, you have what are called terminal cisternae. And the terminal cisternae are what actually attach to the T-tubules at the end of the sarcoplasmic reticulum. When you get two cisternae, so one on the inside on either side of the T-tubule is in the middle, the terminal cisternae are on either side, that forms what is called a triad. So two terminal cisternae plus one T-tubule equals a triad. So of course that is going to equal 3. The sarcoplasmic reticulum is extremely important because it stores and releases calcium ions, which are going to be extremely important when it comes to the muscle contraction itself. If we get a closer look at our muscle fiber, we can see our endomysium surrounding the individual muscle fibers, but then we can see a myofibril being pulled out of the muscle fibers. So what a myofibril is it's a subdivision within the muscle fiber itself. So the muscle fiber is made out of lots of myofibrils. And the myofibrils are actually what are responsible for the muscle contraction itself. And the myofibrils are made out of, they're simply bundles of protein filaments. And what these filaments are, and I'm sure you've all heard of these, are actin and myosin. So you have thin and thick filaments. The thin ones are actin, and the thick ones are myosin. This is a great slide to show you the sarcomere. The sarcomere is the smallest functional unit of a muscle fiber, and it's really important because this is where you have the striating pattern of the myofilaments, or the myosin and the actin, as well as something called titin. Okay, so let's take apart and look at the individual parts of the sarcomere. You have the on either side of the sarcomere, you have what are called Z-lines. And the Z-lines mark the boundary from one sarcomere to the next sarcomere. So this myofibril is being dissected. If you wanted to chunk it apart or cut it apart, you could cut it and get lots and lots of sarcomeres. So this is the perimeter of the sarcomere. Right smack in the middle, you have what is called the H-band, and the H-band is divided by the M line. So within the M line and within the H-band, this is only thick filaments. There are no thin filaments, so no actin within this part. The A-band itself, you mostly see the dark bands and the M line. That's what we're looking at here. The other thing you have are stabilizing proteins that keep the thick filaments in the proper position. If we go outside of the A band, the I band is the light bands, and they're light because they have no thick filaments. So this is only thin filaments. And if you look at the zone of overlap, so the zone of overlap is where you have the actin and the myosin overlapping each other. This is a dark region, and this overlap of the filaments is important for contraction purposes. The other thing I want to point out here is titin. And titin is an elastic protein, and it's going to run from actually the tips of the thick filaments to the Z line itself. So if you can kind of see, it starts at the thick filaments and runs to the Z line, and you can see that on both sides of the Z line. This is important because the titin is going to help keep all the filaments in their proper alignment. It is also important in restoring resting length. So after contraction takes place, it's going to restore the original length of the sarcomere. This is a great shot showing you the individual, the sarcomeres. You can really see the Z lines here delineating each sarcomere. The H band is clearly distinguished. That overlap area, very, very dark. You have, what you're seeing there is the thick strands. The I band, of course, is very light in color. That's because you have no myosin there. That's only the actin. So great shot here. We're taking an even closer look here at the thin filaments, especially the actin filaments. So there are several parts that you need to understand about the thin filaments, and they contain what are called F-actin, or filamentous actin, G-actin, which is globular or globular actin, and nebulin, as well as tropomyosin and troponin. So let's start with the F-actin. F-actin is actually composed of 2 strands of G-actin molecules. So if you want to think about a beaded necklace, let's say you had a pearl necklace, and you took 2 strands of pearls and you simply started twisting them. That's kind of what this is looking like. Okay, so the F-actin strand is composed of the twisted globular molecules of G-actin. And these sites, so the G-actin has specific sites on them that are going to be able to bind to myosin. Okay, so the myosin, of course, is your thick filaments. The G-actin is going to bind to it on these active sites. Then we get to Nebulin. Now Nebulin, if you notice, is kind of in the middle of this twisting strand of F-actin and G-actin. So the Nebulin is going to hold the F-actin strand together. So once you twist it, you have to kind of have that glue to hold it together. That's the job of the Nebulin. Then we get into the two big components that we often think of when we talk about muscle contraction, and that's troponin and tropomyosin. So tropomyosin is going to cover the active sites on the G-actin. This is important because in order for the binding of actin to myosin, they have to have the active sites uncovered. So tropomyosin is going to keep them covered until we get to the position of where we want to contract. And then finally, we get to troponin. And notice troponin is kind of sitting on top of the tropomyosin. This is really important because it is going to bind tropomyosin, G-actin, and calcium ions. And this is going to be really, really important because the calcium, when it binds to troponin, it's going to activate it. It's going to cause that protein to move the tropomyosin to expose the active sites on the G-actin to myosin. Okay, so it's kind of a lot going on there to understand, but we're going to watch this, and you also should see this in the video when you watch the crash course video on muscle, okay, and muscle contraction. So troponin, tropomyosin, they're both proteins. That's important because whenever something attaches to a protein, it's going to change its shape. So that's exactly what's happening here. Now we have to look at the thick strands or myosin strands. So notice here's a better look at the tightened strand that attaches right at the very end of the myosin strand. And remember, that's going to aid in restoring the sarcomere length. So tighten is very important. Now, as we look at the thick strands, these are composed of about 300 myosin molecules. And each of these myosin molecules has a head and a tail. So the whole molecule is composed of the myosin head and the myosin tail. And these are important because we're going to start looking at how these myosin heads attach to the actin at that G-actin active site. At this point, we're going to start talking about what happens during a muscle contraction, and we're going to look at the sliding filament theory. I have posted a very, very short YouTube video under Chapter 10 in that file with your crash course videos. It's only about 3 minutes, not even quite 3 minutes, but it really shows you the details on the sliding filament rule. So please make sure you watch that video in addition, because it'll really help. It's hard to visualize when you're just looking at a static slide, so the animation will help you understand the sliding filament rule. So the first thing I want to look at is just when a muscle is relaxed. You can see that there's no overlap. You don't have, you have a little bit of overlap in the bands at the end of the A band, but what you don't have is a shortening of that sarcomere at this point. As you have a contraction taking place, notice how the sarcomere, the Z lines, actually come together and you have greater zones of overlap. The I band shortens, the A band shortens, and the thin filaments are sliding over or toward the center of the sarcomere. They're kind of drawn in. So if you kind of took your hands around a slinky, right? I think you guys still remember what a slinky is. And you relax it and just kind of let it be, and then you compress it together with both hands coming together. You crunch everything together. That's essentially what happens in a contraction. And notice what overlaps here. That those I bands, they're getting shorter, and you have that intense overlap within the A band section. So how do we get to the sliding filament theory? Well, we cannot do this without the central nervous system telling the muscle, hey, you need to move, you need to contract, you need to pick something up, you need to pull something, you need to do some sort of work. So what happens is an action potential actually travels down the motor neuron from the central nervous system to the peripheral nervous system and attaches at the muscle at the neuromuscular junction. So the junction is named for what it is actually doing. Now, at this neuromuscular junction, you have the neurotransmitter acetylcholine getting ready to be released through the synaptic cleft, and it's going to attach to the receptors on the skeletal muscle. When that neurotransmitter, which is acetylcholine, we know it as ACH, when that attaches at the receptor site, it is going to excite or cause the action potential to take place. Now, how does this happen? We know that an action potential, we have to reach threshold about negative 55 millivolts or so. We have depolarization. The sodium ion channel gates closed, the potassium ion gates open. Then we have repolarization, hyperpolarization, back to resting membrane potential. So you should know the steps of an action potential. You learned all of that in 205. When this action potential travels down into the muscle, how does it do that? It does this along the T-tubules. So the T-tubules carry that action potential deep into the muscle through the muscle fibers. When this happens, it causes the sarcoplasmic reticulum, which is storing calcium ions, to release those calcium ions. This is crucial, okay? This is a crucial step. Those calcium ions are going to bind to the troponin, and when it does that, it's going to make the troponin move the tropomyosin, and when the tropomyosin moves, it's going to uncover the active sites on the G-actin. When the G-actin active sites are exposed, the myosin heads can grab onto the actin. This forms what is called a cross bridge. And this cross bridge is going to enable the myosin to pull the actin closer to the center of the sarcomere, causing a contraction to take place. Now, this cross bridge is really important. It's going to happen all along remember there are 300 myosin molecules, so this is going to happen all the way along this action. When the pulling part takes place, this is called a power stroke. And this is when the myosin head is pivoting to cause the actin to move. This all requires energy. Nothing new there, right? Everything requires energy. So it's going to require energy. The power stroke is going to happen, and then the myosin head is going to release the actin, recock or reposition itself, and then it's going to reactivate to grab on again and pull the actin closer. Okay? When this happens, the muscle is shortening, and this is going to produce tension or pull, we might say. Okay? And when we get this movement or this contraction, this shortening of this muscle, it's important to understand that we are overcoming a load or resistance. So think about a bicep curl. If you put the weight in your hand, in order for you to lift that weight and shorten that bicep muscle, so I want you to put your hand on your bicep muscle, and now I want you to bring up your fist all the way up until you can get your hand as close to your shoulder as possible. What happens to that bicep? You should feel it getting larger or shortening, okay? So you are overcoming the resistance of the weight. Now, the entire muscle is actually shortening at the same exact rate because all the sarcomeres are going to be contracting together. So the speed of the shortening, the speed of the contraction, is dependent on how many power strokes, how many times myosin grabs the actin, pulls it, and then recocks and then pulls it again. So this is important because it depends on a couple of different things. First of all, how much neural stimulation do we have? You have to have the central nervous system telling you what's going on, how much to pull, how much force, how much contraction we actually need. We also have a dependence on the calcium. If we run out of calcium ions, that contraction is done, because as soon as you run out of calcium ions, that troponin goes back and you no longer have exposure of the actin heads. You also have to have ATP available. If the ATP is not available, you've got nothing. No energy, no contraction. So if you run out of calcium, if you run out of ATP, or if the neural stimulus stops, this whole process reverses itself. The calcium goes back to the sarcoplasmic reticulum, the troponin takes the tropomyosin, repositions it so that it's no longer exposed, the neural stimulus stops, and the muscle does what? It relaxes. So it's important to understand the process of a muscle contraction as well as muscle relaxation. Very, very important. I made a note in your lecture notes, just an interesting fact about rigor mortis. This is after a person dies, you have a fixed muscular contraction, so everything tightens up and a person gets very, very stiff. That's kind of where that nickname comes from. Not very nice probably, but you'll remember it, I think. So this happens because the ATP actually runs out and all the ion pumps stop functioning, and the calcium ions build up in the cytosol of the cell, and you just have this constant contraction taking place. So there's your interesting fact for today. In the last slide, we talked about when a muscle is contracted, it is producing tension. Now, the amount of tension is dependent on a couple of different things, but it's important to understand that a muscle is either under tension or contracted, or it's relaxed. There's really not too much of an in-between until we talk about muscle tone. But how do we get the different amounts of tension within a muscle or sarcomere in and of itself? Well, the amount of tension depends on a couple of different things. How much stimulation, how frequently the muscle is being stimulated, and how many power strokes are being performed. So the tension produced, we can look at something called the length-tension relationship. And what happens is the tension of the muscle fiber depends on the length of the sarcomeres. And you actually have different amounts of tension. And this slide is a little bit busy, I know that, but it's showing you where the tension can take place. So you have an optimal resting length. And if you don't get to optimal resting length, you will affect the amount of tension that can be produced at any one given time. So, for instance, if a sarcomere is too far stretched out, the zone of overlap is reduced, and the cross-bridge interactions, you notice they can only interact with a little tiny piece of the actin. So you cannot get as much of a contraction. If the zone of overlap is reduced, then you don't have any, if you look at that next slide on the right-hand side, you can't get any contact. There's no myosin heads able to contact the actin filaments at all, and you get absolutely no tension, no contraction at all. So this is extreme stretching of a muscle, which we don't normally see because we don't allow our muscles to get to this point. Okay, we also can get maximum tension, and this is produced when the overlap is large, but the actin doesn't go over the sarcomere center. So that's when we would get maximum tension, and you can see that this is dependent on optimal resting length. So if you see here, that's really important. The normal range of the length is going to be the percent of that optimal length, enabling you to get to maximum tension. Now, if we go to the other side, so on the right side we have the stretching too far. and it's not allowing us to grab and get that proper contraction. You can go the other side and get what happens if you have too much tension and you've got this contracted muscle. Then you cannot get any tension because you can't go any further. So at short resting lengths, which means you didn't get to the full resting length, then you're going to start to contract before you have optimal resting length obtained. So that's going to cause you not to be able to get as much of a contraction. Also, you can have a problem when the thick filaments are actually engaged at the center or come in contact with the Z-lines. What happens is then the myosin heads can't turn and they can't grab a hold of that actin, and you cannot get tension being produced. So when we talk about length tension, what's important to understand is you really need to get your muscle back to relaxation. There's an optimal resting length, and that resting length has to be obtained in order to get the maximum tension on that muscle. If it's too stretched out, you don't get maximum tension. If it's too contracted, you don't get maximum tension. So don't get too hung up on the length of the length tension relationship. Just understand that there is an optimal resting length. If you don't achieve that, you won't get optimal tension or maximum tension being obtained. Okay, so I probably went on and on with this topic, and I didn't mean to, but I just kind of want you to understand that this is really important. It's an important topic because you have to have that optimal relaxation to get the optimal tension.